Welcome to the Deep Dive, where we plunge into complex information and pull out the most fascinating practical insights just for you. Today, we're taking a bit of a shortcut, actually, into the incredible world of histology, a text and atlas 8th edition. Think of it like having a secret key to unlock the unseen universe inside our bodies.
Mhm.
That's really what histology offers, a way for scientists and doctors to peer into the microanatomy of cells, tissues, organs, and, crucially, understand how their intricate structures dictate their vital functions. So this deep dive is all about revealing the diverse, really ingenious methods histologists use, from basic staining to some seriously cutting-edge microscopy, and why these microscopic insights matter so deeply to your understanding of health and biology.
And that's exactly the point, isn't it? Histology is so much more than just, well, describing what's there. It's deeply connected to molecular and cell biology. The techniques we're about to explore are the very foundation for understanding how every cell, every tissue, every organ actually works. These insights are absolutely critical, not just for groundbreaking scientific research, but also for everyday clinical diagnosis. Think of pathologists identifying diseases, for instance.
Okay, right, let's unpack this then. Before we even get near a microscope, there's a huge amount of work that goes into preparing these tissue samples. You mentioned the workhorse method, the one you'd see first in pretty much any histology lab, hematoxylin and eosin, H&E staining.
H&E, that's the one. Usually after something called formalin fixation.
Fixation, okay.
That's right. The first critical step is fixation. And this is usually a chemical process, most often using formalin, which is basically a formaldehyde solution. Its main job is to permanently preserve the tissue, essentially freezing it in time.
Freezing it in time, I like that. Why?
Well, we do this for a few key reasons. It stops the cells from continuing their metabolism,
Huh?
prevents them from self-digesting, that's called autolysis,
Ah,
kills any microorganisms that might be lurking around, and it hardens the tissue by cross-linking its proteins, makes it easier to handle. And a really neat trick with formaldehyde is that it doesn't significantly change the 3D shape of most proteins.
oh, that's important.
Yes, very important.
Yeah.
Especially for some of the more advanced techniques later on that rely on specific molecular shapes, like immunocytochemistry.
Okay, so the tissue is fixed, preserved like a tiny, delicate artifact. What happens next to get it ready for slicing? Because you can't slice something soft, right?
Exactly. After fixation, the tissue is carefully washed and then dehydrated. Imagine slowly drawing all the water out, maybe like with a delicate sponge, using increasingly strong alcohol solutions, right up to 100% alcohol.
Okay, step by step.
Then comes a step called clearing. We use organic solvents, something like xylene or toluol. These solvents replace the alcohol and prepare the tissue to be infiltrated with warm, melted paraffin wax.
Ah, paraffin wax. Like a candle.
Sort of, yes.
Yeah.
Once this wax cools and hardens, it creates a solid block with the tissue embedded right inside, nice and firm.
Got it. Ready for slicing.
Now for the cutting. We use a specialized machine called a microtome. It's essentially a high-precision slicer. It cuts these wax blocks into incredibly thin sections. We're talking maybe 5 to 15 micrometers thick.
Micrometers, how thin is that, really?
Well, think about a human hair. It's like slicing that hair lengthwise into dozens of even thinner ribbons. It's that thin.
Wow.
These really delicate slices are then floated onto water.
Floated?
Yep, helps flatten them out. Then they're carefully picked up and mounted onto glass slides using an adhesive.
And then finally the color, because otherwise it's just a clear slice of wax and tissue, right?
Yeah.
You didn't see much.
Precisely. You'd see almost nothing. So once it's on the slide, we dissolve out that paraffin wax, and then we rehydrate the section. Back goes the water.
Reverse the dehydration. Okay.
Then it's stained with hematoxylin. Now, this dye is, you can think of it as like a basic positively charged magnet. It's attracted to the negatively charged components in the cell.
Like DNA.
Exactly. Primarily the phosphate groups in DNA and RNA. So it brilliantly stains cell nuclei, and also areas rich in cytoplasmic RNA, this vivid blue-purple color.
Blue-purple for the nucleus. Got it.
After that, we dehydrate the slide again and then stain it with eosin. Now, eosin is the opposite. It's an acidic, negatively charged dye. So eosin loves the positively charged components.
Like proteins?
Yes, like the amino groups of proteins. So it stains most of the cell cytoplasm and the extracellular fibers like collagen, a vibrant pink or red.
So you get blue nuclei and pink cytoplasm.
Generally, yes.
Yeah.
That combination gives you this stunning contrast, painting a detailed map of the tissue.
Makes sense.
Then just a few final steps.
Yeah.
Dehydrate it again. Clear it usually with xylene, mount it in a non-aqueous medium, and add a cover slip. That makes a permanent preparation ready to view. It's really the bedrock of diagnosis because it lets pathologists quickly spot abnormalities. The patterns just jump out.
So H&E is the bread and butter, the foundation. Gives us that essential overview. But you mentioned it has limitations. It doesn't show everything.
That's right, great question. H&E is fantastic for general structure, but it doesn't show everything. For example, routine processing with those organic solvents. It dissolves neutral lipids, fat droplets basically. They just wash away.
Oh, so you can't see fat with standard H&E.
Not easily, no. If we want to see those, we typically use frozen sections.
Frozen? How does that work?
Well, the tissue is rapidly frozen, say in liquid nitrogen or cold isopentene, very quickly. And then it's sliced while still frozen in a special refrigerated microtome called a cryostat.
Okay, a cold slicer.
Yep. And these frozen sections can then be stained with specific dyes that actually dissolve in fats, making them visible. And this approach, this frozen section technique, has a really critical clinical correlation.
Oh, how so?
Well, frozen sections are often a surgeon's best friend during an operation.
Really? During surgery?
Absolutely. Imagine a surgeon removing a tumor. They need to know immediately if they got all of it, or if the edges are clear. They'll send a tiny tissue sample, maybe from the margin, straight to the pathology lab.
While the patient is still on the table.
Exactly. Within as little as 10 minutes, sometimes even less, that sample is frozen, sliced, quickly stained maybe with H&E or a faster stain like methylene blue. And a pathologist makes a preliminary diagnosis.
10 minutes? That's incredible.
It is. And it allows for immediate, on-the-spot decisions right there in the operating room. It can save crucial time, potentially avoid the need for a second surgery. It's hugely impactful.
Wow. Okay, that's a powerful application.
Mm.
What about other fixatives or stains?
Well, for electron microscopy, which gives us, much higher resolution,
Right, the really zoomed-in view.
we need even more specialized fixatives. Something like osmium tetroxide is essential. It binds beautifully to phospholipids, the main components of cell membranes. It preserves those delicate membrane structures and adds electron density, which makes them visible under the electron beam. Without it, membranes are hard to see in EM. And beyond H&E, there are loads of selective stains. For instance, orfusin or resource and fusin highlight elastic fibers. Silver impregnation methods are great for reticular fibers and basement membranes. Tiny structures H&E often just glosses over.
So we've gone from preserving tissue to basic staining to specialized stains for specific molecules like fats or fibers. How do we push deeper into histochemistry and cytochemistry to really understand the chemical makeup and maybe the function of cells?
Now we're moving from just seeing shapes and colors to understanding the underlying chemistry and activity. That's histochemistry and cytochemistry. But first, it's important to realize what actually remains in a routine H&E section after all that processing.
Right, because you said some things get lost.
Exactly. What we mostly see are the large insoluble molecules, nucleoproteins like DNA, cytosolital proteins, extracellular proteins like collagen, membrane complexes, the big structural stuff. But many crucial components are actually lost. Small soluble proteins, transfer RNA, those neutral lipids we mentioned, glycogen often gets washed out, proteoglycans, glycosamidoglycans, all the soluble bits like ions and metabolites are gone.
So knowing what's missing is as important as seeing what's there.
In a way, yes. It helps you correctly interpret what you are seeing and know the limitations.
Okay, so back to the chemical basis of staining.
Right. Just to reiterate, acidic dyes like eosin, pancreate, love positively charged, or basic components, they show acidophilia. Basic dyes like hematoxylin, blue-purple, love negatively charged, or acidic components like nucleic acids, they show basophilia.
Acid loves base. Base loves acid. Got it.
But here's a fascinating twist called metachromasia.
Metachromasia. Sounds interesting.
It is. Certain basic dyes, like toluidine blue, which is normally blue, actually change color when they bind to certain specific molecules called polyanions, things with lots of negative charges packed together.
They change color. How?
The dye molecules essentially stack up or aggregate when they bind to these polyanions, and this aggregation shifts the color they absorb and emit. So the blue dye might turn red or purple.
Where would you see that?
Oh, places like the matrix of cartilage
Wow.
or inside mast cell granules. It's a really useful visual clue that tells you something specific about the chemical nature of that area.
Cool, like a built-in chemical test?
Kind of, yeah. And speaking of chemical tests, another really powerful one is the periodic acid shift reaction, or PAS reaction.
The PAS, okay.
PAS, stained carbohydrates and carbohydrate-rich molecules, a really distinctive magenta color.
Magenta for carbs.
Yep. It's great for things like glycogen granules inside cells, mucus in goblet cells, the basement membrane underlying epithelia, and those reticular fibers we mentioned.
How does it work chemically?
Well, the periodic acid step creates aldehyde groups on the sugar rings, and these aldehyde groups then react with the Schiff reagent to produce that magenta color. It's quite specific.
And you can use this quantitatively. You mentioned something about DNA.
Yes, it's Fulgen microspectrophotometry. It uses the same Schiff reagent principle, but after a specific acid hydrolysis step that only creates aldehyde groups on DNA.
So it stains only DNA magenta.
Correct. And because the reaction is stoichiometric, meaning the amount of color is directly proportional to the amount of DNA, you can actually use a microspectrophotometer to measure the intensity of the stain in individual nuclei and quantify the DNA.
Why would you want to quantify DNA?
Oh, it's incredibly useful clinically. Pathologists use it to study DNA changes in dividing cells, maybe during cancer development, or to analyze ploidy, the number of chromosome sets in malignant cells. Abnormal ploidy patterns can give crucial information for evaluating tumor prognosis and predicting treatment response.
That's powerful stuff from a simple stain reaction.
It really is. We can also use enzyme digestion as a control.
A control? How?
Well, say you think a PAS stain is showing glycogen. You can take an adjacent, identical section and treat it first with an enzyme like diastase, which specifically breaks down glycogen. Then you do the PAS stain. If the magenta staining disappears compared to the untreated slide, you've confirmed it really was glycogen.
Clever, like erasing the target.
Exactly. And then there's enzyme histochemistry itself. Here, the goal is to pinpoint where specific enzymes are active in the tissue. Usually, you don't stain the enzyme itself because there might not be much of it. Instead, you provide the enzyme with its substrate and capture or visualize the product of the reaction it catalyzes, right where it happens.
So you see where the enzyme works, not where it is.
Often, yes. For example, you can detect acid hydrolases, which are typical of lysosomes, or ATPase activity in cell membranes, which tells you about active transport. It gives functional information.
Okay, this is getting really specific. Now, what about pinpointing individual proteins or even specific gene sequences within a cell? That sounds like the next level.
Absolutely. That brings us to immunocytochemistry, or ICC. This technique is built on the incredible specificity of the immune system.
Antibodies.
Exactly. The highly precise lock and key fit between an antigen, the target molecule you're interested in, and an antibody designed to recognize it. We take these antibodies and tag them, maybe with a fluorescent dye for light microscopy, or an electron-dense marker like gold particles for electron microscopy.
So the antibody finds its target, and the tag lights it up.
Precisely. We have two main types of antibodies used. Polyclonal, which are a mixture recognizing different parts of the target molecule,
Okay.
and monoclonal antibodies. These are amazing. They come from a single, identical cell clone, so every antibody molecule is exactly the same, targeting one very specific site or epitope on the antigen.
Monoclonals sound very precise.
They are. And they have absolutely transformed medicine. They're used routinely for pinpoint tumor diagnosis and typing. They can distinguish different types of lymphocytes, for instance, and they're even used therapeutically for targeted drug delivery, guiding drugs directly to cancer cells.
That's huge. And how does the visualization work, directly tagging the main antibody?
That's one way, called the direct method, but more common is the indirect method.
Indirect.
Yeah, you use an unlabeled primary antibody that binds to your target antigen. Then you come in with a secondary antibody that's tagged, say, with fluorescence, and is designed to bind specifically to the primary antibody.
Why the extra step?
It amplifies the signal. Several secondary antibodies can bind to each primary antibody, making the target much brighter and easier to see. It increases sensitivity.
Ah, clever amplification. Okay, so that's proteins. What about DNA or RNA sequences, genes?
For that, we turn to hybridization techniques. The principle here is using complementary base pairing, A with T, G with C, and DNA. We create probes, short sequences of nucleotides that are complementary to the specific DNA or RNA sequence we want to find. These probes are labeled somehow.
Like molecular fishing lines with hooks.
That's a good analogy. You introduce these probes to the prepared tissue section, and they will hybridize or bind specifically to their complementary target sequence if it's present.
How do you see where they've bound?
A fantastic example is fluorescence in situ hybridization, or FISH.
FISH. Okay.
Here, the probes are labeled with different fluorescent dyes. This allows you to visualize multiple different DNA sequences simultaneously, often on chromosomes, using different colors.
Multiple colors. Wow.
Yes, it's a game changer in clinical genetics. It's used extensively for detecting chromosomal abnormalities like extra chromosomes in Down syndrome or deletions or translocations associated with certain cancers, often even in prenatal diagnostics using fetal cells.
Incredible diagnostic power.
Definitely. Then there's another classic technique called autoradiography.
Autoradiography sounds like
Yeah.
self-pictures with radiation.
Sort of. It's like creating a molecular treasure map using radioactivity. Scientists introduce precursors like amino acids or nucleotides that have been tagged with a radioactive isotope.
Okay, radioactive building blocks.
Exactly. Cells take these up and incorporate them into newly synthesized large molecules like proteins or DNA. Then, you coat the tissue section with a photographic emulsion, like old school film. You leave it in the dark for a while. Wherever the radioactivity is concentrated in the tissue, it exposes the silver grains in the emulsion directly above it.
So you develop it like a photo and see black dots where the new molecules were made.
Precisely. It shows you exactly where those new molecules were synthesized or accumulated. It was crucial for figuring out many metabolic pathways.
Fascinating. Now, you mentioned something really cutting edge, expansion microscopy, making the sample bigger. How on earth does that work?
Right, expansion microscopy or ECSM. It sounds like science fiction, but it's real. The core idea is to physically enlarge the specimen itself to overcome the resolution limits of traditional light microscopes.
Physically enlarge it.
Yes. You infuse the fixed tissue with chemicals that form a swellable polymer network, a hydrogel, actually similar to the stuff in baby diapers, right within the sample, anchoring the biomolecules to it. Then you digest away the original tissue proteins that would resist swelling, and you add water. The hydrogel swells up, extending isotropically uniformly in all directions.
How much bigger?
Standard protocols get about a 4.5-fold linear expansion. That's over 80 times in volume. And iterative methods can go even higher, maybe 20 times linearly.
Things that were too close together get pulled apart.
Exactly. Molecules or structures that were closer than the 250 nanometer resolution limit of a standard light microscope get physically separated. Now they are far enough apart to be resolved by that same conventional microscope. It effectively gives you super resolution, maybe down to 25 nanometers or even better using standard equipment.
That's ingenious. Just make the target bigger.
It is. And it's led to a new field called expansion pathology, where this technique allows pathologists to see nanoscale details relevant for diagnosis, things that previously might have required much more complex and expensive electron microscopy.
Mind-blowing. Okay, it's astounding how much goes into preparing these slides and developing ways to reveal their secrets. But now, let's shift focus slightly to the incredible instruments themselves, the microscopes. What does all this prep mean for how the microscopes actually work?
Well, the fundamental concept for any microscope is its resolving power.
Resolving power, not just magnification.
No, magnification just makes the image bigger. Resolving power is the ability to show two closely positioned objects as separate, distinct images. That's the real measure of a microscope's quality.
Okay, so how good of it?
Think about it. Your naked eye can distinguish objects that are about 0.2 millimeters apart. Maybe a bit better for some people. A good bright field light microscope, however, can resolve down to about 0.2 micrometers.
Micrometers again.
That's a 1,000-fold improvement. It's the difference between seeing a blurry dot and seeing individual cells or even large organelles inside them. The objective lens is what determines this resolution primarily. The ocular lens just magnifies the resolved image.
1,000 times better. Got it.
And here's a crucial point, especially for students starting out. Organs are three-dimensional complex structures, but our histological sections are almost always two-dimensional slices.
Right, the thin slices we talked about.
Yes. And mentally reconstructing that original 3D structure from looking at multiple flat 2D images is a key skill, but also a common challenge.
Yeah, I can imagine, like seeing only one slice of a cake.
Exactly. Imagine slicing an orange. A slice through the middle looks different from a slice near the end. The book uses a great example with a kidney structure, renal corpuscle. Depending on where you slice through that ball of capillaries, the 2D image looks completely different.
So you need to look at many slices, or just understand the 3D possibility?
Both, really. Recognizing this helps avoid misinterpretations. And speaking of misinterpretations, we also need to be constantly aware of artifacts.
Artifacts, like errors.
Yes, basically defects introduced during any stage of the preparation, fixation, embedding, sectioning, staining, maybe a wrinkle in the section or stain precipitate or shrinkage. They aren't real biological structures, and learning to recognize them is critical.
Okay, good point. So the most common microscope.
That would be the bright field microscope. It's the standard you'll find everywhere. It's pretty straightforward conceptually. A light source at the bottom, a condenser lens to focus the light onto the specimen on the stage, the objective lens for primary magnification and resolution, and the ocular lens or eyepiece for final viewing.
And staining is key here.
Absolutely essential for bright field, because most unstained biological material is translucent and offers very little contrast. The stains provide that contrast, making things visible.
Okay, but there are ways to see unstained things.
Yes. Beyond basic bright field, there are several other specialized optical systems that are really clever. Phase contrast and differential interference contrast, often called DIC or Nomarski optics. These are amazing because they do let us examine unstained specimens, even living cells.
Living cells? How?
They exploit tiny differences in the refractive index, how much light bends as it passes through different parts of the cell. They convert these invisible phase differences into visible differences in brightness or contrast.
So you can see structures inside a living cell without staining it.
Yes. It's fantastic for observing cell behavior, movement, division. Nomarski optics are particularly good at giving a pseudo-3D, almost bas-relief appearance, highlighting surface textures.
Cool. What else?
There's dark field microscopy. In this setup, the condenser blocks the direct light path. Only light that is scattered by tiny structures in the specimen actually enters the objective lens. So small particles appear as bright specks against a completely dark background.
Like dust motes in a sunbeam.
Exactly like that. It's very useful for visualizing things that are too small or hard to stain properly, like certain bacteria, such as spirochetes.
Okay. And fluorescence, we mentioned that with staining.
Right. Fluorescence microscopy is incredibly important research today. You illuminate the specimen with UV light, or a specific wavelength of visible light, and fluorescent molecules, fluorophores that you've introduced, maybe tagged antibodies or fluorescent proteins, absorb that light and then emit light back at a longer visible wavelength, making specific structures glow brightly.
Like tiny beacons inside the cell?
Precisely. And building on that is light sheet fluorescence microscopy, or LSFM. This is really powerful for imaging larger, thicker, often transparent specimens. Instead of illuminating the whole sample, it uses a thin sheet of laser light to illuminate just one focal plane at a time.
Like optically slicing it.
Exactly. You move the sheet or the sample and capture a stack of these optical slices, which you can then reconstruct into stunning 3D images of labeled structures deep within tissues, often with minimal photo damage.
Great for developmental biology. Wow. More ways to see 3D. What about confocal? I hear that term a lot.
Confocal scanning microscopy is another sophisticated technique for getting sharp 3D images. Its trick is using a pinhole aperture placed in front of the detector. This pinhole physically blocks any light that comes from outside the precise focal plane you're interested in.
So it rejects the blurry, out-of-focus light.
Correct. Only the crisp, in-focus light gets through the pinhole to the detector. You scan a laser beam across the specimen point by point, plane by plane, collecting light only from that tiny focal spot each time. Then a computer reconstructs the image, slice by slice. It gives incredibly sharp optical sections, free from the haze you get in conventional fluorescence microscopy, allowing for detailed 3D reconstructions, like a CT scan for cells.
Okay, super sharp optical sections
Good.
and super resolution, going beyond the normal limits.
Yes. Super-resolution microscopy represents a whole family of techniques designed to push past that 200-250 nanometer diffraction limit of conventional light microscopy.
Breaking the rules.
Kind of. They use clever tricks. For example, single-molecule localization methods like PALM or STORM involve photo-switching individual fluorescent molecules on and off, pinpointing their exact centers with much higher precision than the diffraction limit would suggest, and building up an image over time. Another approach is STED microscopy, which uses a second laser beam shaped like a donut to effectively switch off fluorescence around the central excitation spot, making the spot much smaller than the diffraction limit.
The result.
Resolution's down to maybe 10 or 20 nanometers. It allows us to see cellular structures like the detailed organization of synapses or cytoskeletal elements in unprecedented detail, sometimes even in living cells. It's truly revolutionized cell biology.
Incredible detail with light. But for the ultimate resolution.
For the ultimate resolution, we still generally turn to electron microscopy, or EM.
Using electrons instead of light.
Exactly. Electrons have much, much shorter wavelengths than visible light photons. This allows EM to achieve resolutions perhaps 1,000 times greater than light microscopes, down to the sub-nanometer scale in some cases, revealing the ultrastructure of cells.
1,000 times better again. So what types are there?
Two main types. First, the transmission electron microscope, or TEM. Here, a beam of electrons passes through an extremely thin specimen section, much thinner than for light microscopy, maybe 5100 nanometers. Different parts of the specimen scatter or absorb electrons to different degrees, especially after staining with heavy metals like lead or uranium to add contrast. This creates an image on a fluorescence screen or digital detector, showing internal ultrastructure, organelles, membranes, viruses.
Internal details. Got it. What's the other type?
The scanning electron microscope, or SEM. SEM is different. Instead of passing through a focused beam of electrons, scans back and forth across the surface of the specimen. This knocks secondary electrons off the specimen surface, and these are collected by a detector to build up an image point by point as the beam scans.
So it sees the outside.
Yes, it produces these amazing, often breathtakingly detailed, three-dimensional looking images that show surface topography. Think of those incredible pictures of insects' eyes or pollen grains or the surface of cells. That's usually SEM.
Right, those almost sculptural images.
Exactly. And one more, which is quite different, atomic force microscopy, or AFM.
Atomic force, it sounds intense.
It measures tiny forces. It's unique because it's not optical or electron-based at all. It uses an ultra-sharp physical probe, like a tiny record needle, but much sharper, attached to a flexible cantilever. This probe literally taps or drags across the surface of the specimen. As the probe encounters bumps and valleys on the surface, the cantilever deflects. A laser beam reflecting off the cantilever measures this deflection with incredible precision, allowing a computer to map the surface topography.
It feels the surface, down to atoms.
Potentially down to molecular or even near atomic resolution in some cases. And a huge advantage, especially for biology, is that AFM doesn't require a vacuum like EM does. It can image specimens, even living cells, in their natural aqueous environment under physiological conditions.
Feeling living cells. That's remarkable. So there you have it. What a journey. A rapid but, I think, fascinating tour through the incredible methods behind histology, from preparing a seemingly simple H&E slide, learning about frozen sections, specialized stains, immunochemistry, FISH, all the way to the cutting edge of expansion microscopy, super-resolution light microscopy, EM, and even AFM. Each technique really does peel back another layer of complexity, doesn't it? Helping us understand that intricate dance between structure and function at the microscopic level. It truly is a journey from the invisible to the profoundly insightful.
Indeed it is. And what's truly compelling, I think, is how each new method, each innovation in staining or microscopy, builds upon the last. It allows us to ask more precise questions, to probe deeper, and gain much richer insights into the fundamental units of life cells and tissues. It's this constant quest for better resolution, for more specific information, and for clearer understanding, which ultimately enhances our clinical knowledge and our diagnostic capabilities.
And that pursuit of clarity, that deeper understanding, is what this deep dive is all about for you, the listener. We really hope you've had some aha moments today, and maybe feel a bit better equipped to appreciate the incredible, intricate, invisible world within us, and also appreciate the ingenuity behind the tools we've developed to see it with such stunning clarity.
You know, this raises an important question, thinking about the future. Given the incredible detail these methods can now reveal about microscopic anatomy, what specific aspect of cellular or tissue structure may be revealed by one of these advanced techniques do you think holds the most significant potential, the most exciting implications for future medical breakthroughs? Is it understanding synapses better? Or maybe the extracellular matrix? Something to perhaps ponder on your own.
That's a great thought to leave our listeners with. Until next time, keep exploring the incredible insights that emerge when we take a deep dive into your sources.